We will continue our exploration of a real-life experiment that went wrong. This episode forms the second part of the fourth and final troubleshooting experiment. Yes, this is going to be the last experiment in the story. The experimenters did not know it at the time, but I would like to let you know so that you will have an end in sight and you will have a better understanding of the story. We will discuss the remaining practical challenge of the story that we mentioned in the last episode. Then we will talk about how the experimenters dealt with the challenge, how they designed the final troubleshooting experiment, what they found, and how they ultimately solved the original problem in the pilot experiment. We will also see how they revised the original protocol for successful experiments going forward. Again, the experimenters showed mental dexterity, that is, the careful thinking process. And we can learn from that. Hi everyone, welcome to the Foundational Skills in Life Sciences podcast. I'm your host, Dr. Synaptologika. We discuss the skills that life science scholars learn, use, and improve throughout professional activities. Your benefit is expressed in my motto: learn the skills, enjoy academic life. Last episode. We are in a long mini-series of reading a life science paper. Today, we are in episode 24. If you are a new listener of my episodes, please start listening from episode 7. It was the start of the mini-series of reading. We are continuing to discuss a real story. It is a story about one specific pilot experiment that did not work well at first. We are discussing it because it will tell us that negative controls are important when we design life science experiments. It will also tell us that negative controls are important when we detect potential problems in experiments and when we troubleshoot them, always together with the positive control counterpart. The experimenters were trying to test if an active form of a specific chemical compound stimulates the cultured cells so that their function would increase. For this purpose, they wanted to find that the active form would increase the function in comparison to the inactive form of the same compound, which was expected not to increase the function. The problem the experimenters faced was that the cell function was already increased from the baseline level even before they treated the cells with either an active or an inactive form of the compound. The cell function had been expected to stay at the low baseline level from the start, up to the time when the cells were treated with the chemical compounds. So the cell function had been stimulated erroneously, somehow and somewhere earlier in the protocol. To solve this big problem, the experimenters designed and performed 3 troubleshooting experiments, as we have seen in the previous episodes. They found that the cell function was low at the end of the first week culture period. During this period, the cells were cultured and maintained in the old culture medium that contained animal serum. They also found that the cell function was high during the 1-hour treatment period, even when the cells were not treated with any chemical compound. Based on these findings, the experimenters narrowed down the problem to be in the intermediate period that they called the stabilization period. It was between the first culture period and the third treatment period. This stabilization period was the time when the old culture medium was replaced with the fresh culture medium, and the cells stayed there for 1 hour in preparation for the chemical treatment and for the functional measurement. In the last episode, the experimenters came up with a hypothesis. The hypothesis was that the problem might be an unintended change in the compositions of the culture media, and it may have stimulated the cells unknowingly. They refined it and expressed it as the updated idea 4. It states that removing the serum from the culture medium immediately led to a sustained, maximal increase in cell function. They knew that the original problem could be explained if all parts of this idea were correct. So the next thing that the experimenters wanted to do was to test this idea by designing and performing the fourth troubleshooting experiment. Then I posed a question to you. I said that the experimenters were very close to designing the troubleshooting experiment, but they were not fully ready yet. They noticed a crucial fact, and the fact presented one more practical challenge to solve. Do you know what the challenge would be? I also gave you a hint. Pay attention to how to change the media practically. Did you come up with a good idea? Today's episode. Let's start by discussing the practical challenge that the experimenters had to overcome. Then we will talk about how the experimenters tested the updated idea 4 in the fourth troubleshooting experiment. This was in fact the last troubleshooting experiment in our story. What the practical challenge was. The goal of the fourth troubleshooting experiment is to test the updated idea 4 that said, removing the serum from the culture medium immediately led to a sustained maximal increase in cell function. So some people might suggest going back to the original protocol and recording the cell function before and after the medium exchange. If the cell function is increased after the medium exchange, a part of the updated idea 4 will be proved to be correct. But is this true? Let's review the medium exchange in the original protocol. There were two types of culture media. At the start of the stabilization period, the medium was exchanged from the old medium with serum directly to the fresh medium without serum. Do you notice that two factors were changed at the same time when the media were exchanged? One factor was the serum. When the medium was exchanged, the serum was removed. This is what the experimenters wanted to test. But there was another factor. Another factor is the refreshing. The old medium was exchanged with the fresh medium. In other words, the old culture medium was refreshed. Then it is possible that something in the old medium could be lost when the old medium was exchanged with the fresh one. The lost materials could be, for example, materials released from the living cells during the culture period. And such a loss can change the cell function. These two factors were changed simultaneously in the original protocol. If a cell function was changed by this medium exchange, was it because the serum was removed? Not necessarily. A change in the cell function can be caused by one of the three possibilities. 1. Removing the serum alone. 2. Refreshing the media alone. Or 3. Both removing the serum and refreshing the media. Multiple potential causes for a single effect. The experimenters will not be able to pinpoint exactly which possibility can explain the functional change. This will be a bad experimental design. This was the practical challenge that we talked about in the last episode. The practical challenge was that if the experimenters exchange media as in the original protocol, they would not be able to answer clearly if serum removal stimulated the cell function. This was because the experimenters would not be able to evaluate the effect of serum removal separately from another factor, which was refreshing the medium. I had repeatedly stated that the old culture medium with serum was replaced with the fresh medium without serum. So the media refreshing has been stated as a fact throughout our episodes. But whether we pay attention to the fact is another thing. The fact has been presented repeatedly. What could be missing is the realization that this media refreshing could serve as another potential factor that increases the cell function, in addition to serum removal. How to solve the practical challenge. Here the experimenters wanted to test if serum removal increases the cell function. As we have just seen, the original protocol was not good enough. It would not allow the experimenters to separate the two factors. What was specifically wrong with the original protocol then? The original protocol was wrong for the test because it exchanged the media in a single step. In other words, the two factors were changed in a single step. Serum removal and media refreshing happened at the same time. Then, how to solve this practical challenge? The solution would be to exchange the media not in a single step, but in two steps. And at each step, only one factor would be changed. This way, the effect of serum removal from the media can be isolated from the effect of refreshing the media, and they will be analyzed separately. Now, do we agree that we always want to have a clear vision like this about what to analyze and how to analyze it? I think that the experimenters did a wonderful job. They realized that two factors were changed by a single-step media exchange, and they solved this practical challenge by changing the protocol to a two-step media exchange. This was very good. How to implement a two-step media exchange. The next step was to think about how to change two factors independently by implementing a two-step media exchange into the protocol. 3 types of media and how to exchange them. What the experimenters did was to use 3 types of media so that they would be exchanged in 2 steps. Superficially, their design could look more complex than necessary, but as we have just discussed, the 2-step media exchange will make it possible what a single-step exchange of 2 media types is not able to. Let me explain the details. Among the 3 types of media, the first and the third media were the same as in the original protocol. The second medium was the new type. It will serve as an intermediate type. Here's how to exchange 3 types of media in 2 steps. 1. Initially, the cells were bathed in the old culture medium with serum. This was the first of the 3 media types. This was the medium at the end of the culture period. 2. By the first media exchanging step, it was exchanged to a fresh medium with serum. This was the second of the 3 media types. In this first media exchanging step, the old medium was changed to a fresh medium, but the serum was present throughout at the same concentration. So, by monitoring the cell function before and after this media exchanging step, the experimenters could evaluate the isolated effect of refreshing media on the cell function while the serum was present. 3. Then, by the second media exchanging step, the second medium was changed to a fresh medium without serum. This was the third of the 3 media types. In this second media exchanging step, the serum was removed while the base culture medium remained fresh throughout. So, by monitoring the cell function before and after this media exchanging step, the experimenters could evaluate the isolated effects of removing serum on the cell function using the fresh culture media. So the key was to introduce the second type of culture medium, that is the fresh medium with serum. By using this intermediate type, the experimenters could isolate the 2 factors, that is, refreshing the media and removing serum. This way, the experimenters could evaluate the effects of those 2 factors on cell function independently of each other. I would say, what a good design! Comments about how to remove serum from culture medium. Here, I would like to give you 8 comments. In these comments, I would like to clarify what we mean by removing serum from culture medium. I would also like to give further comments about 2-step versus 1-step media exchanges and about why fresh culture medium was necessary. Comment 1. First, the basics. What is serum? Serum is the liquid part of the whole blood that remains after blood clots have formed and the blood clotting factors and blood cells are removed. Serum is composed of more than hundreds of chemical components. Those components include electrolytes such as sodium, potassium, calcium, and chloride, and sugars, proteins, lipids, and much more. The actual types of components will increase as our understanding proceeds from now, and the actual number of components will increase as the sensitivity and reliability of the detection mechanisms will increase from now. Comment 2. Can we remove serum from culture medium physically and completely? It would have been the simplest approach if the experimenters could physically remove serum from the old culture medium. If it is possible, the cultured cells will be bathed in the culture medium with serum, as before, and then the serum will be physically removed from the same medium. Then the cell function can be analyzed before and after serum removal, with the base solution being the same, that is, the old culture medium. But removing any solid or liquid component from a liquid solution is very difficult. I would say it is almost impossible. For example, suppose you have added sugar to your coffee and you have mixed it well. After that, can you remove the added sugar? It will be very difficult to remove all the added sugar without removing any other components of the coffee, like the liquid part of the coffee and a small amount of natural sugar that was contained in the coffee from the start. To make matters worse, the serum is composed of more than hundreds of chemical components, so it is practically impossible to remove all serum components from the medium physically and completely. Comment 3. Then, how did experimenters remove all components of the serum practically? Instead of trying hard to remove all components of the serum one by one, the experimenters resorted to a very common and practical approach. In a simplified scheme, it is to prepare two media, one with serum and the other without serum. And the medium with serum that bathes the cells will be exchanged with the medium without serum. This will effectively remove the serum from the media. So when we say removing the serum from the medium, it practically means exchanging the medium with serum to medium without serum. Comment 4. Some people might ask, can we still enforce a single-step media exchange for assessing the effect of serum removal? The answer is that a single-step exchange is practically difficult. In principle, a single-step media exchange is possible when the experimenters change the protocol a little. In the original pilot experiment, the cell function was measured from the cells bathed in the fresh medium without serum. But what if the cell function is to be measured from the cells bathed in the old medium without serum? Then the experimenters would need to prepare only two types of old culture media. One is the old culture medium with serum. This is the culture medium that bathes the cells during the culture period, and we have been talking about it. The other type is the old culture medium without serum, which is new here. These two types of old culture media would be enough for evaluating the effect of removing serum on cell function, and they would need only a single step to exchange media. There is no need to analyze the effect of refreshing the medium. This idea sounded good. Unfortunately, it was not practical. The reason is that it is not easy to prepare an old culture medium without serum. To prepare such a culture medium, the experimenters would need to culture the cells without serum, separately from the main culture with serum. They would retrieve the old medium without serum from this separate culture, and then they would use it for replacing the old culture medium with serum in the main culture. But the reality is that those cells do not grow well when they are cultured without serum. So the old culture medium without serum in the separate culture would contain less of the materials normally released from the cells than the old culture medium with serum in the main culture. The result will be that the two types of old culture media, with and without serum, would be different. And this situation would be similar to the two types of culture media, old and fresh. Then the protocol would need two-step media exchanges to separately analyze the effect of removing serum, as we have just discussed. So, a single-step media exchange was not an attractive option. Comment 5. Some of you might be wondering why the experimenters wanted to use fresh culture medium in addition to the old culture medium in their protocol. In the following 3 comments, I would like to discuss why it is important to use fresh culture medium, especially for measuring cell functions. Comment 6. Before discussing fresh culture medium, let's briefly review what we discussed about the old culture medium. In episode 20, we described what we mean by old medium and the advantages and disadvantages of it. An old culture medium is the medium already used for cell cultures. So the old culture medium will change from the unused fresh medium during usage. And the change will reflect the fact that the old culture medium has been in contact with the various stages of cells, that is, the growing, mature, dying, and dead cells. Here are details of some features. One of the advantages of the old culture medium is that the living cells would have secreted some materials into the solution, and those materials could be beneficial for cell growth, maintenance, and for other purposes. One of the disadvantages of the old culture medium is that the nutrients have been consumed by the cells, so that not much could be remaining in the solution. Another disadvantage is that the solution would contain dying and dead cells, and the materials released from those cells would be bad for further use of the medium. There's one more disadvantage of the old culture medium. We will talk about it in the next comments. Comment 7. Here, we would like to give a similar comment to fresh culture medium. Advantages and disadvantages of using fresh culture medium are the mirror images of the old culture medium. One of the advantages of the fresh culture medium is that the nutrients have not been consumed by the cells yet, so that it is rich in nutrients. Another advantage is that the solution does not contain dying and dead cells, which could be bad for the cell culture. But one of the disadvantages of the fresh culture medium is that it does not contain the materials that the living cells will secrete into the solution, so that it might not be ideal for cell growth or maintenance. Comment 8. In addition, the fresh medium has one key advantage, and correspondingly, the old medium has one disadvantage. It is how well or poorly the experimenters can control the medium composition. As we have discussed, the composition of the old culture medium is affected by the living, dying, or dead cells. So the composition will be variable depending on slightly different culture conditions. Here, I would be happy to give 2 examples of variable parameters that could affect the medium composition. One parameter is the number of cells in a culture container, a culture dish, a culture plate, or a culture flask. The higher the cell number is, the higher its impact is on the medium composition. Another factor is the degree of water evaporation. Let me explain. The cultured cells are kept in a culture container. The container is usually small enough to be held in one hand. The container is placed in a culture incubator, which is like a small piece of furniture at home, something that looks like a refrigerator. Here is one fact. The culture medium inside a culture container is loosely connected with the air in the culture incubator. This allows the control of the culture medium, such as the levels of oxygen and carbon dioxide, by controlling the levels of oxygen and carbon dioxide in the incubator air. mammalian cells are kept in a culture incubator. In the incubator, the temperature is maintained usually at 37 degrees Celsius, which is near the body temperature, and the humidity is maintained at 100%. But the water in the culture medium evaporates during the culture period. This should not happen because the humidity in the incubator is supposed to be maintained at 100%. But it does happen, maybe because we open the incubator door to do some work on other cultured cells. When we open the door, even for a short period of time, the humidified air leaks out of the culture incubator, so the humidity goes below 100%. After the incubator door is closed, the humidifier in the incubator will start working to increase the humidity to 100%. But during this time, the water in the culture medium will evaporate. What I have described is my simple reasoning. If the humidity setting is below 100% within the incubator, this will surely lead to evaporation of water from the culture medium. There could be more causes of evaporation. Anyway, the fact is that water evaporates from the culture medium. A single culture plate has multiple separate wells under a single lid, each well containing a set of cultured cells. If the number of wells is, for example, 24 or 96, there are wells that face the outer edge of the culture plate, and there are wells that are away from the outer edge. Very often, the wells at the outer edge of the culture plate tend to have more water evaporation than the wells in the middle of the culture plate. As the water evaporates from the culture medium, the concentration of all the materials in the culture medium will increase. So different wells in a single culture plate will experience different degrees of water evaporation, and so different concentration of all the materials in the medium. Why are we talking about the variability under these culture conditions? This is because variable conditions will lead to slightly different compositions of the old culture media among different cultures or different wells in a single culture. These variable conditions will create variable environment for the cells, and it is not appropriate for functional measurement because the variable environment can affect the functions themselves. For example, a different concentration of a simple ion, potassium ion, will lead to different resting membrane potentials of all cells, which in turn could affect the functions that an experimenter will try measuring. One key disadvantage of old culture medium is this variable condition. In contrast, the fresh culture medium has a well-controlled composition because it is freshly prepared and it has not been used yet. This is the key advantage of the fresh culture medium. This is the reason why the experimenters wanted to use the fresh culture medium for functional measurements. Professor's viewpoint. Please let me give you still another comment here. I have talked about some practical details of cell cultures. My goal is not to train you in how to do cell cultures. Cell culture is just an example of a wide topic. Instead, my goal is to help you develop mental dexterity so that you will approach life science experiments and interpret them with flexibility and an openness to multiple possibilities. I would not want you to think dogmatically. For example, let's say you set the humidity controller of culture incubator at 100%. Then please do not think that the humidity in the culture incubator should be maintained always at 100%. It is possible that the humidity controller is not calibrated well. Then, even if you set it at 100%, it is possible that the humidity will not achieve 100%. Also, the humidity within the culture incubator can fluctuate from time to time, as we have discussed. These practical aspects are often overlooked, but practical considerations can be critical for success of certain experiments. It will be helpful to keep these and other possibilities in mind. That's my 2 cents. what we have discussed so far today. So far today, we have talked about 3 things. They were seemingly complicated, but all of them turned out to be necessary. They will help us understand the design of the 4th and final troubleshooting experiment. The first thing we talked about was the practical challenge that the experimenters had to overcome. It was how to separately analyze the potential effects of 2 factors, serum removal and refreshing media. The solution to the challenge was to exchange 3 types of culture media in 2 steps, rather than to exchange 2 types of culture media in 1 step. And in relation to the very practical procedures, we talked about the second thing, which was what we mean by removing serum from the culture medium. When the cells are bathed in a medium with serum, removing the serum practically means to replace the medium with serum by the medium without serum. And the third thing was the advantages and disadvantages of using fresh culture medium, and why fresh culture medium is necessary for recording the cell function. Fresh culture medium was necessary because it allows better control of the cellular environment than the old culture medium. The composition of old culture medium is harder to control, and so the old culture medium will experience slight variations in cellular environment. They could change the cell function unknowingly, which is not good. Design of the 4th and final troubleshooting experiment. Now we are finally ready to discuss the design of the 4th troubleshooting experiment. Let me remind you again. The goal of this experiment is to test the updated idea 4. It said, removing the serum from the culture medium immediately led to a sustained maximal increase in cell function. The 4th troubleshooting experiment included 4 groups. The first group was the positive control. After the stabilization period, the group was treated with an active compound during the treatment period. This group was the same as the original positive control. The second group was a new positive control. It was treated similarly as the original positive control, but with a single difference. The difference was that the concentration of the active compound was 2-fold higher than the original concentration in the first group. By comparing the cell function in this new positive control with the original positive control, the experimenters can determine if the cell function was maximally stimulated in the original positive control. The third group was the mock treatment negative control. After the stabilization period, this group was treated with an inactive compound during the treatment period. This group was the same as in the original experiment. And lastly, there was the fourth group. It was the no treatment negative control 4. This group did not go through the stabilization period, and this group was not treated with any chemical compound of interest. This group was similar to the no treatment negative control 3 in the third troubleshooting experiment, where the cell functions were measured only once, and the cell functions were measured in the old culture medium without changing the medium. But the no treatment negative control 4 in the new group had 2 differences from the no treatment negative control 3. One difference was how many times the function was measured. In the no treatment negative control 4, the cell function was measured not once, but multiple times at different time points. This way, the experimenters could learn the time course of functional changes. Another difference was a major one. In this no treatment negative control 4, the culture media were exchanged. Remember that the goal of the fourth troubleshooting experiment was to remove the serum from the culture medium and to examine its effects. To do that, the experimenters prepared 3 types of media and exchanged them in 2 steps, as we have discussed earlier today. The 3 media were: 1. The first medium was the old culture medium with serum. This medium was the same as in the original experiment. This was the medium that bathed the cells during the culture period. In this experiment, the functional measurement was started while the cells were still bathed in this medium. 2. The second medium was the fresh culture medium with serum. So in the first step of media exchanges, the old medium was changed to a fresh medium, but the serum was present throughout at the same concentration. The second medium is a new solution. The difference between the two media was that the first medium was old, whereas the second medium was fresh. The functional measurements were continued in the second medium. So, by monitoring the cell function before and after this media exchanging step, the experimenters could evaluate the isolated effect of refreshing media on the cell function while the serum was present. 3. The third medium was the fresh culture medium without serum. So in the second step of media exchanges, the fresh medium with serum was changed to the fresh medium without serum. The third medium was the same as the medium during the stabilization period in the original experiment. Both the second and the third media were fresh media. The difference between the two media was that the second medium was with serum, whereas the third medium was without serum. Function measurements were continued in the third medium. So by monitoring the cell function before and after this media exchanging step, the experimenters could evaluate the isolated effect of removing serum on the cell function using the fresh culture media. Comment 9. This will be a short comment about the functional measurement. In all the experiments that we have discussed today, the function could be measured only once from a single set of cells. This specific function could not be measured continuously in real time. This feature was determined by the nature of the measurement method. So, for example, this means the following. If the experimenters want to measure the cell function at 4 time points, they have to prepare 4 separate sets of cells, and each set would be measured only once at a specific and different time point. If they want to measure functions at 4 time points and from 3 sets of cells at each time point, they will need a total number of 4 times 3 equals 12 sets of cells. Now, let's go back to the main content. What were expected if the updated idea 4 was correct? If the updated idea 4 is correct, then there would be at least 3 expectations. The first expectation is related to part 1 of the updated idea 4. It asks, when does the function change in relation to the media exchanges, and what is the causative mechanism? The expectation is that the cell function will be unaffected when the medium is refreshed in the presence of serum, but the cell function will be increased immediately when the serum is removed from the fresh medium. Reflecting this expectation, the no treatment negative control 4 is expected to show the following 3 findings when the 3 media are exchanged in 2 steps. 1. Low cell function in the old culture medium with serum. 2. Low cell function after the culture medium is refreshed, still in the presence of serum. And 3. High cell function after the serum is removed from the fresh medium. The second expectation is related to part 2 of the updated idea 4. It asks, how long does the functional increase continue to last? The expectation is that the functional increase will last well over 2 hours after serum removal. Where does this expression, over 2 hours, come from? It came from the protocol of the original pilot experiment. There, the cell function was measured after the 1-hour stabilization period and 1-hour treatment period, so well over 2 hours after the start of the stabilization period when the culture media were exchanged. Reflecting this expectation, the no treatment negative control 4 will practically show the sustained high cell function well over 2 hours after serum removal. The third expectation is related to part 3 of the updated idea 4. It asks, is the functional increase maximal and therefore saturated? the expectation is that the cell function will be increased maximally and saturated after the serum is removed. Reflecting this expectation, the new positive control, which will be treated with the active chemical compound at twofold higher concentration, that is at the supramaximal concentration, that will not show higher function than the original positive control treated with the maximal concentration. In other words, the cell stimulation will be saturated at the maximal level. In addition, the no treatment negative control 4 will show an increased function to the same high level as these two positive controls. What they found with the fourth troubleshooting experiment. The results of the fourth troubleshooting experiment were exactly the same as the expectations from the updated idea 4 that we have just talked about. So all parts of the updated idea 4 were proved to be correct. The experimenters were able to examine different parts of the idea separately and completely because they thought deeply about many aspects of the experiments, and they created a systematic and excellent design of the troubleshooting experiment. This was gorgeous. At this point, the experimenters felt satisfied and they could stop the series of troubleshooting experiments. Side note. Here's a side note. A minor side note. Do you know the movie Back to the Future? I know this is an abrupt turn of a story, but I wanted to tell you about this movie. It is a 1985 classic American science fiction film. Dr. Emmett Brown is a mad scientist. He was called Doc, and his pet dog was named Einstein. I like this mad scientist, Doc. He had invented a time machine, but a special fuel was missing. He had to find a method to energize the defunct flux capacitor, the dynamic engine that enables the time machine to bring Marty, the protagonist, the principal character, back to the future. Then suddenly Doc obtained a bright idea that a lightning bolt in the sky would be able to energize it. Then he exclaimed, "This is the answer!" If Doc was in the experimenter's team in our story, he would have said the same thing when he solved all the problems. This is the answer. Well, I like him. If you find my name, Dr. Synaptologica, to be too long, you can simply call me Doc. Let's go back to the main content. New protocol. After all the troubleshooting experiments, the experimenters edited and upgraded their original protocol so that the experiments would work well in their laboratory. Let's check the new protocol. The experimenters changed one point in the protocol. In the original protocol, the medium contained the serum only during the culture period. In the new protocol, the medium contained the serum at the same concentration throughout the experiment. In other words, the serum was present not only during the culture period, but also during the subsequent stabilization period and the treatment period, and during the functional measurement. Stabilization period was still necessary. This was the time after the culture period, when the old medium was replaced with the fresh medium, and when the cells were not stimulated. There are two reasons for why the stabilization period was still necessary. The first reason was that it allowed the cells to keep a relatively stable and low baseline level of function. Without the stabilization period, the cell function fluctuated to some extent, and it made the measurements a little unreliable later in the protocol. The second reason for keeping the stabilization period in the new protocol was that it was necessary to replace the old medium with the fresh medium. As we discussed today, the fresh medium will allow the experimenters to control the medium composition so that the cell function can be measured under a well-controlled, uniform condition. Design of the experiment based on the new protocol. The design of the experiment remained the same as the original pilot experiment, except for one change in the protocol that we discussed just now. The serum was now present in all the media at the same concentration throughout the experiment. there were only two groups as in the original experiment. One, the positive control treated with an active form of a chemical compound, and two, the mock treatment negative control treated with an inactive form of the same compound. Using this design with the upgraded protocol, the experimenters successfully found the expected result for a pilot experiment. The active compound increased the cell function, whereas the inactive compound did not. After the successful pilot experiment, the experimenters could examine the effects of other chemical compounds as test groups, as they had planned in the first place. All the experiments worked well in the end. This is it for today. Here is my plan for the next 2 episodes. In the next episode, we will reflect on what we can learn from this story about a series of troubleshooting experiments. There are many things that we can learn, and they will be helpful in any life science field. After that, we would like to have an episode for 1-year anniversary of my podcast. Your action. Until we meet next time, please think about what we have learned in our story. If you are not sure, one focus can be on negative controls. When you get an idea, please send me an email about it. We can think about it together. My email address is ideas@synaptologica.com. Thank you for joining me in today's episode of Foundational Skills in Life Sciences. I'm Dr. Synaptologica. I'll see you soon. Bye for now.